but he left that and he's with us here only to deliver a talk, and he has promised this to you as well as to me also. For that purpose, I am very much thankful to Ashish sir, and I requested him to continue with his talk, which is on biofertilizer and biopesticide chemicals. Over to you, sir.
Thank you, Dr. Fayaz, for giving me this opportunity to interact with you on the very important topic of this biofertilizers and biopesticides and overall the role of biopesticides in today's agricultural scenario. As you are aware that at the national level, the government of India is interested to align the agricultural research and other activities as per the norms of organic farming and natural farming. And with the passage of time, it is expected that the importance of these inputs will be on a rise, and they will be very much needed in the field, in the research, and everywhere as the country will progress towards the organic farming. Though organic farming cannot be done on a very large scale in respect of food grain crops, but in some niche crops and high value crops, it definitely has a role to play to improve the economic condition of the farmers and overall to improve the environmental quality and the particularly with respect to the changing climate. Organic farming, natural farming, alternate farming, low zero budget farming, these are all actually the synonymous terms where the final objective is to produce quality food on a sustainable manner and protect our environment and whatever damage we have done to the environment to reverse to that damage to some extent. As you are aware, the other day, 2 days you might have spent in the conference itself, the aims and objectives of the conference were to deliberate on the issues that are related to the climate change. And climate change has been induced due to the anthropogenic activities, particularly the industrial activities, and a significant portion comes from the agricultural activities also. The other day, I was preparing uh, this talk for one of the VIPs of the university, and while going through the literature, I learned that 14% of the gases, greenhouse gases, are contributed by the agriculture. And overall, if you combine it with the other activities, that means when we produce the food, that is the pure agricultural activity, primary agricultural activity. When we transport and process it, that is the secondary agricultural activities. When we combine the entire amount of greenhouse gases from all these activities, from production to processing and then reaching that product to the consumers, the entire activities contribute 30% of the greenhouse gases. So you can imagine the importance of agriculture and allied industries in contribution to the greenhouse gases and deteriorating the environment, global environment. So we have to do all that is required to reverse those changes so that the at least we can restore it to the era where we started, say, to the era of before 1950. In the last 70 years, 60 to 70 years, the carbon dioxide concentration has gone to up to around 412 ppm. And the temperature since industrial revolution has increased by 1 degree Celsius, average temperature. And that is, it seems that 1 degree Celsius is nothing, but it is huge when you perceive it from the GDD point of view. It adds a lot of calories, these growing degree days to the plants, to the animals, and puts a lot of stress on the ecosystem in terms of the more evaporation from the soil, more transpiration from the plants, changing pest and disease scenario, changing of the weed scenario, more disasters, more these what we call as the extreme weather events. Those includes floods, flash floods, droughts, cold spells, heat waves, hailstorms, hurricanes, windstorms, and many more. Even these flash floods then also trigger landslides. And the other day, our honorable vice chancellor was telling rightly that our Himalayan agroecosystem is very fragile to all these extreme weather events, because you know the slope is very, very high in the mountains. Even small amount of rain, intense rain, can trigger erosion, landslide, deforestation, and many more damages, right? So let us straightway come to the topic of this biofertilizer carriers. So you may have, since your graduation, you may have gone through this definition. The biofertilizers are actually preparations containing live or latent microbes which help in enhancing the soil fertility either by fixing atmospheric nitrogen, solubilization of phosphorus, or decomposition of organic wastes, or by augmenting plant growth by producing growth hormones with their biological activities. So the association of the rhizobium with the legumes is one aspect. If you augment, apply to the seed the rhizobial cultures, then that will increase the efficiency of the nitrogen fixation, because there are many strains of a particular microbe. Some rhizobial strains may not be much efficient, and others may be very efficient. So you have to augment with the efficient strains, isolate them from the plants, and then use them again. Then we have other group of organisms that are important in solubilization of the phosphorus from the native soil phosphorus. How they do it? Actually, these bacillus and other many pseudomonas species, they secrete organic acids. When the pH slightly falls in the soil, it results in the solubilization of the this native phosphorus, whether it be inorganic or organic phosphorus. So we have a lot of phosphorus in the soil, but that's not available. If you do something about it using these microbes to solubilize a fraction of phosphorus, then you may be saving a lot of resources. Then we are also using, like trichoderma and many other fungal species, we are also using it to decompose organic matter in a faster way. Otherwise, the organic matter from these crop residues or other materials of plant origin, it takes a lot of time to decompose. If you enhance that decomposition process, that will help us in releasing more nutrients in a faster way for the plants. Then we have another group of organisms which are called as plant growth promoting organisms that release certain hormones, and those hormones stimulate the growth of the crop plants and increase the yield, and even help the plants to evade stress, to resist stress, to tolerate stress. So this is short about the biofertilizer definition. Then these groups we have discussed. For example, rhizobium, it's a symbiotic association. Then azotobacter is a free living bacteria that can function in open soil without the plant support itself, but it requires organic carbon as a food source. Then we have azoospermum. It lives in association with the plant roots, but not inside the roots, but very close to the plant roots. Then we have blue-green algae. These are actually cyanobacteria that are very important in the certain aquatic environments. For example, in rice, we use blue-green algae as the biofertilizers. Then we have higher plants such as azolla. Azolla also fixes atmospheric nitrogen. It is association between a fern and a cyanobacteria that helps in fixing the atmospheric nitrogen, producing a lot of biomass. It's a green manure and it's a source of nitrogen. So that azolla is generally used in tropical environments where rainfall is high, where soils doesn't dry up, where temperature is neither too low nor too high. We have also certain species in Kashmir. In Punjab also people have tried it, but under subtropical and temperate conditions, it has not been a much success because of certain climatic reasons. In South India, when you will see, say Kerala, there is a lot of rainfall. Temperature doesn't go too high beyond 35 or 38. Then it stays and produces a lot of biomass. They use it as a feed to the cattle as well as to the poultry also. But when you go to Punjab in the summer, there is a lot of feed. Field is dry, so it won't produce much biomass. In Kashmir, when you come, it doesn't produce much biomass because of low temperature. So we have to identify, it is onus lies on the microbiologists to identify the species that can work under those conditions. Then phosphorus solubilizing bacteria we have discussed. Then compost enhancer. There are a lot of fractions which are very recalcitrant to decomposition, such as certain types of cellulose. Then we have lignin. Then certain complex polysaccharides. Then there are certain microorganisms, particularly the actinomycetes, certain fungi that are able to degrade those recalcitrant type of fractions that are present in the plant tissue. So use of those in the compost pits can enhance the decomposition process and make the available compost in a shorter span of time. Then phospho means that solubilize the phosphorus. Rhizo means nitrogen fixation in the nodules. Then azo means fixes atmospheric nitrogen near the roots. Then we have tricho, non-pathogenic, eco-friendly, antagonistic, hyperparasitic against different pathogens and biocontrol agents. I think the last one you are knowing much better than me, because for the last 10 days you might have heard more about this trichoderma, which is used widely not only as a pesticide but also for faster decomposition of the different types of plant materials while preparing the compost. So rhizobium is for the leguminous crops. Azotobacter can be used in wheat, rice, vegetables as a free living organism. Azoospermillum lives more in association with the rice and sugarcane, maybe in maize also. Then blue green algae, because it's an aquatic microbe, those live in the rice. Rice, we have to keep a lot of water in rice. Then we have azolla. Azolla also needs a lot of water, so it is compatible with rice. Then phosphorus solubilizing microorganisms or other plant growth promoting microorganisms are suitable for all types of crops. Then potential of biopesticides. About 1 lakh tons of biopesticides are produced in the agriculture in India annually. The intensive use of pesticide in agriculture is a cause of concern. The problem is especially serious because of development of resistance to pesticides, important pests and presence of pesticide residue in the food. Then growing pesticide resistance has meant that large proportion of agricultural production is lost to the pests. Then hence alternative environment friendly method of plant protection are required. Because of these use of pesticides, there is a lot of problem that contaminates our food and environment and induces the resistance in different types of pests. And this is also happening in weeds. When you use a lot of herbicides against the weeds, it induces resistance. Similar thing happens in insects, even in the fungi and bacteria. When you use a lot of antibiotics or fungicides, then certain strains, through natural selection, through selection pressure, they become selected and they produce, and then that antibiotic or that pesticide or fungicide or herbicide doesn't act, is ineffective or isn't no longer effective. So so what are these biopesticides? Biopesticides are derived from animals, plants, and microorganisms such as bacteria, fungi, and viruses. They are inherently less harmful than the chemical pesticides. They are more target specific than chemical pesticides, affecting only the target pests and their close relatives. In contrast, chemical pesticides often destroy friendly insects, birds, mammals, etc. So they are often effective in small quantities. So doses are also low. Then Bacillus thuringiensis, I think you might in your UG and PG courses, you might have heard about this bacteria, how it acts and how it kills the insects. When BT releases toxins inside the midgut of the pestis, it kills. It kills because it has a certain types of proteins it produces, and that results in the death in the plant. So Bacillus is one of the biopesticides. Then we have baculoviruses, and how they act. These baculoviruses has been depicted diagrammatically here. These target specific viruses which can infect and destroy a number of important plant pests. They are particularly effective against lepidopteran pests, cotton, rice, and in cotton, rice, and vegetables. Large scale production poses certain difficulties, so their use has been limited. So at production front, it is very difficult to produce these baculo type of viruses. On a smaller experimental level or on a smaller scale, they are used in IPM modules. So then we have neem. So this is simply an oil that is extracted from the leaves as well as from the seeds of Azadirachtica, and this nimidin and many other products that are present in this neem have insecticidal properties. And there are many uses of this neem. Even it's used as a nitrification inhibitor. It has been added to the urea now, neem oil. The urea pack would come in 50 kg bags. Now it is 45 after this covering, after covering the urea granules with this neem oil. it has nitrification inhibiting properties. When nitrification is slowed down, it increases the nitrogen use efficiency. That is why the weight of 50 kg bag of urea has been reduced to 45 kg. At least that means it is 10%, it increases the efficiency by 10%. And that is significant amount. So now this trichoderma, as the Fayaz Saab might have, definitely he would have elaborated on all the aspects of the trichoderma. A fungicide is a fungicide effective against soil-borne diseases and root rot. It is particularly relevant for dry land crops such as groundnut, black gram, green gram, chickpea, which are susceptible to these diseases. In India are marketing these trichoderma products. And now we should also try this one in rice, particularly with respect to the Bakani disease. I think Fayaz Saab and the breeders are already doing something about it, and see whether it can act in those saturated conditions. Then biofertilizers are usually prepared as carrier-based inoculants containing effective microorganisms. Incorporation of microorganisms in carrier material enables easily handling, long-term storage, and high effectiveness of fertilizers. Before we would produce those liquid biofertilizers, previously it was entirely carrier-based. A solid carrier was used as the base, and then we would inoculate it with the relevant strain, and then we would store it properly, and then we can easily transport it. But now the more of the focus is being has shifted to the liquid biofertilizers. We will discuss why it has happened like that. Then carrier material, various types of materials are used for preparation of seed inoculant. The carrier material should have a size of 10 to 40 micrometers. So that is important. And this size is more efficient. Then properties of the good carrier material, how the carrier material should function and what should be its important properties. First of all, it should be non-toxic to the inoculant bacterial strain. It should be very neutral and not affect negatively these strains. Then it should have good moisture absorption capacity. 40-50% moisture it should hold. Otherwise, when it becomes dry, then it will result in the death of the spores and cysts. Then easy to process and free of lump forming. It shouldn't form lumps. When we store something, say a fertilizer, it forms lumps after absorbing the moisture. So that should not happen with this kind of carrier. Then it should be easy to sterilize. Whenever we produce any biofertilizer or biopesticide and take a carrier should be totally sterilized. Why? पैथोलॉजिस्ट उसके बगैर ब्रीडर बताएंगे, पैथोलॉजिस्ट नहीं बताएंगे. आप बताओ. Why we should, for example, I have shown you the biochar or lignite or peat. There are a large number of carriers. Why should we sterilize it? सबा अगर contamination हो जाएगा तो क्या हो जाएगा? Yeah, it will replace the effective microorganisms because the other microorganisms, we don't need them. They will consume the nutrient, entire nutrient in that carrier, and they will replace the efficient strain, and then we no longer need that unwanted bacteria. So it should be pure, it should be sterilized, and then we have to load the relevant strain on that particular. So that's why we need the sterilization process. And there are different methods, particularly the autoclaving and gamma radiation. Then it should be available in adequate amounts. Then good adhesion to the seeds. It should stick to the seeds, so as when the seed germinates, then it should this spore should also germinate. And in fact, for example, if it is rhizobium, when it sticks to it, then it will come in contact with the germinating seed. Otherwise, it will be lost. Then good pH buffering capacity. It should be around neutrality and non-toxic to the plant as well. So these are the important properties. Then other essential properties of the carrier materials. When we inoculate seed material with this, say we keep it for 1 or 2 days, it should survive on the seed surface. It should survive there also. It should have ability to survive on the seed surface. Then it should tolerate to some extent drying. When we shade dry after inoculation, that slight drying at least it should tolerate. And then the survival of inoculant bacteria in the soil. Then ultimately we have to sow the crop, the seed in the soil. It will take a few days to germinate. Till then also, for a few days, it should survive in the soil conditions also. So that is after application. Then After being introduced into the soil, the inoculant bacteria have to compete with the native soil microorganisms. Again, it should have the ability to compete with the native that are present in the soil, and such carrier materials that offer available nutrient or are habitable micropore to inoculant bacteria will be desirable. So some kind of nutrient also should be there. For example, when we mix lignite with some organic material, say vermicompost, then vermicompost will give the nutrient, and the lignite or the peat or biochar will form the base. It will provide the micropores for inhabiting the bacteria or other strains. So इस तरह से एक दोनों का जैसे literature, I have gone through the literature and I saw that a combination of the both the types of materials gives a better product and more efficient product, and that has longer shelf life. What happens actually when you inoculate the relevant strain with this say a base material, say lignite or peat, and then when you also add the nutrient, then the it starts multiplying inside it. If the moisture and other temperature conditions are normal, then it starts multiplying, the population may go up. So it gives a better condition when you add the nutrient as well as the base material. Then in this sense, materials with microporous structure such as soil aggregates and charcoal will be good carrier materials for inoculant. Then peat क्या होता है? That is coconut peat, वो peat नहीं है. Peat is actually जैसे कोल बनता है ना, ऐसे ही अगर किसी जगह पे lot of biomass is there on the soil surface and then it is slightly partially decomposed. अगर boggy soil कहीं पे देखी होगी lot of biomass under water, तो फिर उस तरह से for a long time it stays there, it turns black. It has some part of soil also inside it. That is actually the peat. This is coconut peat. Peat is actually the initial stage of decomposition, and later when it is subjected to further pressure and time, then it converts into lignite and other materials. We will see the process of formation of the coal and other materials in nature. So the different materials are peat, lignite, perlite, charcoal. Then we have dry granules, composted sawdust, agriperlite, expanded clay, kaolin, silite, diatom, porosil, MP, microsil, and vermiculite. Large number of materials have been tried as the carriers for the biofertilizers, and the nature of these carriers will open on another subject. So you have to take some good book or see the literature from the internet and see what are the different properties of these materials and how best these can be used for the as carrier materials for biofertilizers and biopesticides. So sterilized oxalic acid as industrial waste is used as a carrier. Then alginate perlite, dry granule, composted sawdust, agriperlite, expanded clays, and the relevant bacteria that can be used as that can be used as carrier. Cheese wheat grown cells in peat, mineral soils, even soils and clays are also can be used as carrier. Then coal, then granular inoculants amended with nutrients, soybean oil or peanut oil added with lyophilized cells, then perlite, wastewater sludge, wheat bran, sugarcane bagasse, nutrient supplemented pumice. These are all the carrier materials that have been found suitable for certain bacterial strains, and for certain conditions they can be used. The characteristics have been written in the third column. You can go through, I will give you this presentation, you can go through these slides and see which carrier is better for which strain and what are its important characteristics, right? So this is exhausted a little bit. Then what is coal? This is actually the black gold found in sedimentary strata. Then contains carbon, volatile metal, moisture, mostly used for power generation. Then coal reserves are 6 times greater than the oil reserves. Then we have carboniferous coal. Most of the world's coal was formed 350 million years ago. Then in terms of absolute time, the carboniferous period began 358 million years ago. So you might in your earlier classes you might have seen this kind of thing, how the coal was formed. Actually, in the marshy lands, the huge biomass became submerged and covered under the earth. Then further biomass was produced that were also covered by the earth. Then it went to the deeper layers, and under this intense pressure, it got this biomass under those conditions got converted into the coal, and that coal is now that's actually we are using as the fuel. Then what is peat? Peat is the first stage of transformation. ये peat जो आप उसको बोलते हो, this peat is different. That is coconut peat. वो उसका husk होता है, and that is simply organic material. But this is carboniferous material that has been produced millions of years ago and is under the earth. First stage of transformation, we get the peat. It contains 40 to 55% carbon, then contains sufficient volatile matter and a lot of moisture. Then we have left to itself, it burns like wood. Then we have lignite, lower grade coal, 40 to 55% carbon, intermediate stage, then dark to black brown, moisture content 35%. Lesser moisture it has, it's lignite. Then we have bituminous coal, other form of coal. It has 40 to 80% carbon, higher amount of carbon on average. Then moisture and volatile content 15 to 40%, does not have traces of original vegetable material. The earlier farmers may have some organic material which is still undecomposed. Then we have anthracite. Best quality coal is the anthracite. You see it is shiny black and 80 to 95% carbon, very high amount of carbon. Then we have zeolites, mineral substances, microporous aluminosilicates that have also been tried as carrier materials. They have porous structure that can accommodate wide variety of cations and anions. Then natural zeolites from volcanic rocks are obtained. So these are other materials which you can go through the slides to learn more about it. Then before using any carrier material, we have to sterilize it. And sterilization, as you rightly said, that it will eliminate all the contamination and all the other microorganisms that we actually do not need. Otherwise, they will compete with our carrier strain, this inoculant strain, and they will replace it. So sterilization carrier material is essential to keep high number of inoculant bacteria. Gamma irradiation is the most suitable way of carrier sterilization because the sterilization makes almost no change in the physical and chemical properties of the material. For example, we will take vermicompost, vermicast, or any organic matter, say rice husk. If we boil it, if we autoclave it, it will change its structure, it will change its nature. But this gamma radiation doesn't change the physical properties. It only kills the contamination and the bacteria and fungi, whatever spores will be there. In brief, carrier material is packed in thin-walled polythene bag and the gamma irradiated with 50 kggy. ये यूनिट है इसका जो गामा रेडिएशन का, आपको तो पता है, म्यूटेशन वगैरह आप पढ़ते हैं, ये यूनिट इसका है रेडिएशन, हाउ मच डोज वी हैव टू एक्चुअली एडमिनिस्टर. Another way of carrier sterilization is autoclaving, then keeping the bags in the autoclave for 60 minutes and 121 degrees Celsius. So this is the common and easy method for the laboratory, because gamma radiation everybody cannot use. You have to have certificate to deal with those radioactive materials. Normally the cobalt radioactive material is used for this gamma radiation, but everybody cannot use it in laboratory. You need to have permission from Atomic Energy Commission or whatever agencies are there in Government of India. So what is the principle actually? When gamma radiation hits an atom, it ionizes it. For example, we have radiation in the mobile also, radiation in our radio system also, but that is non-ionizing radiation. But this kind of radiation, because of its energy and its wavelength, it ionizes the biomolecules inside these. And then when it releases the electron, the ionization results in the death of the microorganism. But here you should remember that when there are spores present, spores or cysts, the efficacy is less. But when the cells are vegetative stage, that means when the cells are physiologically active, they will be more of the number of cells will be killed when we put these gamma radiations on them. So the stage in which the microbe is present at that point of time will actually depend. You see both the graphs are depicting that kind of The dose has also been given there. With the increasing dose, the more death will be there, and with the increase in number of vegetative cells, physiologically active cells, better will be the sterilization process. So they will become physiologically active only when you will provide the nutrient and right amount of moisture. Am I right, sir? When we provide them moisture and nutrient, then they will start multiplying and they will come in the vegetative phase. So the effects of radiation on the microorganisms, how we do it: prepare appropriate amount of carrier material, 10 kilograms, divide in 10 kilograms into 10 bags of this much size, 0.1 millimeter, then seal the packages using heat sealer, and then carrier is highly dry, wet with an appropriate amount if it is dry. So moisture also affects. So it should be a bit moist when you put gamma radiations in those materials to sterilize them. So this is the whole setup that is used for the sterilization process. I think Fayaz Saab may be knowing more about it because I'm not a microbiologist, how they do it and what are its principles and how it then is effective to sterilize the material. So these are the effects of radiation on the microorganisms. I think this has been repeated or no. Then the whole process: prepare 1 gram of carrier samples, non-irradiated, and mix with 9 ml of sterile water to make suspension. Dilute the suspension by serial 10-fold dilutions using sterile water. Incubate at 30 degrees Celsius and count bacterial colony number. So this is how we can then see the efficacy of that when we inoculate it. Then storage temperature has been given here, and how to inject the culture in that sterilized carrier material, and how to store it for a longer period of time and better efficacy. Then liquid biofertilizers, as we were telling, in Japan, probably the scientists discovered that liquid biofertilizers are liquid formulations containing dormant form of desired microorganisms and their nutrients, along with the substances that encourage formation of resting spores or cysts for longer shelf life. The dormant forms, on reaching the soil, germinate to produce fresh batch of active cells. These cells grow and multiply by utilizing carbon in the soil and from the root exudates. You see, there is an interaction between the root exudates and the flora that multiplies inside the soil. So plants are in a close interaction with the different types of the this microflora that's present in the soil. Without plant growth, the soil will ultimately become sterile. It won't multiply because there will be no longer organic matter. When we grow plants in the soil, it adds root biomass. Actively growing plants also secrete exudates, and when the death occurs, the decomposition results in the release of the carbon and other nutrients for the microbial growth. So there are several advantages of liquid biofertilizers. Then longer shelf life, no contamination, no loss of properties due to storage at 45 degrees Celsius up to 45, greater potential with native populations. High populations can be maintained at 10⁹ cells per ml up to 12 months. Easy identification by typical fermented smell. Cost saving, carrier material, pulverization, neutralization, sterilization, and packing. Quality control protocols are easy. Better survival of the seeds and soil. No need of running the biofertilizer production units throughout the year. Very much easy to use by the farmer. Dosages are less, 10 times less than the so that is why the carrier based materials are becoming now less popular, and the liquid bioformulations are becoming more popular.
And important is shelf life. You just see
Yeah.
longer shelf life, 12 to 24 months.
12 to 24 months as per the literature. And then the storage temperature and the conditions under you keep it will also definitely affect the overall storage life. Then very high enzymatic activity under these liquid formulations. Then there are 3 ways of using the liquid biofertilizers. You may use it for the seed. You may directly use it in the soil. You may also dip the seedlings inside for some time so that the microbes come on the root surface. Seed treatment, root dipping, and soil application. The entire procedure has been written here. It is for your information. You can go through this presentation. I will hand over it to you and see how these are different. But most of the time we use as the seed treatment because we need less quantity, and then when it directly comes in contact with the seed, definitely that kind of thing will be more efficient for the plants. And Root dipping is only possible when we sow the crop by transplanting. Otherwise, for example, in rice or vegetables, we do transplanting, but in most of the field crops, we go by direct sowing. In that case, the most important procedure is to go by seed treatment. So this is serial dilution a little bit, and the units you might be have learned this already. For example, in this experiment, vermicast has been compared with the charcoal, and you see the in vermicast the population, and you also see the population in the charcoal. What is the trend? What does the trend say? In the last, you will see the overall the vermicast is better. But you have to sterilize the vermicompost. We cannot use the raw vermicast and inoculate it with the bacterial strain or whatever you want. You have to sterilize it through autoclaving first, and then you can use this vermicast as a carrier material. You see, it is only because of the nutrients that the vermicast is providing to this strain, and the population has slightly decreased, but it has drastically decreased in case of charcoal. Then this is another graph which shows the use of crushed cob, compost, biogas slurry, and zeolite. And in this case, the Pseudomonas putida, serratia, then Pseudomonas fluorescens, and many species have been used on these carriers. Then with the time also, they have seen the population, how the population has behaved, active population has behaved. Carrier-based inoculation further improved the efficacy of multi-strain consortium inoculation and increases the growth and yield of physiological parameters of wheat. Then, while in the field conditions, significant increase in the root and shoot dry weight. In this case, the biogas sludge has proved as the better material that has given more shelf life and more number of cells after sometimes say 3 months. So that's a significant finding from this paper. Then another is liquid biofertilizers, azotobacter or morphological parameters in amaranthus. Then here there are 3 treatments and 1 control. One is liquid form of azotobacter, one is carrier based azotobacter, and combined form of liquid and carrier based azotobacter. So in when you combine both kinds of things, when you apply the liquid biofertilizer as well as the carrier based biofertilizer, then the efficiency has been shown as the better. So sometimes the hybrid of the both, when you do both kinds of inoculations, you apply liquid biofertilizer as well as the carrier based. In this case, amaranthus leafy vegetable has been tested in Tamil Nadu, and they saw that when we combine both the carriers, both the formulations, and apply it on this amaranthus, then the results have been found better.
Actually, Doctor, here we can say powder-based formulations.
Powder-based?
Yes, not carrier. Why so? Because in liquid-based formulations also, we use let it be distilled water as a carrier. Okay?
Right.
And stock culture as beta-dextrose brown. Best is to say powder-based formulations as well as liquid-based formulations.
Okay.
Why they might be confused because during the past 15 days, I am telling them you have to develop the formulation in 2 parts. One stock culture and second carrier.
Carrier.
In both, let it be powder-based formulation, you have to develop it on a stock culture and use powder as a carrier. Let it be cow dung, let it be any other vermicompost as a carrier. What you are suggesting here, sir. In liquid-based formulations also, you have to develop a stock and use liquid as a carrier.
Okay.
So don't confuse here. Sir is saying carrier-based means powder form.
Powder form, solid
Solid
form,
powder
form.
formulation. And liquid is some
Definitely, whenever you use the carrier, you cannot use lumps. You have to micronize it first. I told you the size that is 10 to 40 micrometers. So that size can be achieved only when you convert any carrier material into a powder. And in biofertilizer production units, I have seen the very big crushing mills. जैसे क्रशर होता है ना, ये just a minute. Little bit of data I have collected just to show here that sometimes we have when you combine 2 carriers, then maybe the efficiency is improved. That's what the point I want to make here. And the second, then whenever you add a carrier to the soil, for example, a carrier like biochar or a charcoal, it adds more carbon to the soil. So that way also it is beneficial, right? So in this slide, the workers have seen the survival rate of Azospirillum brasiliensis in different ratios of carrier materials, vermicast and lignite. Lignite is a form of coal. We in the earlier slide we discussed lignite, that peat, and then advanced form of coal, and here the lignite and vermicast has been used in different proportions: 0, 1, 1, 1, 2, 1, 3, 1, 4, 1, 5, 1, 6, 1, and 1, 0. And you can see in the last column, when they used the vermicast only, the efficiency was better. The survival rate was better. You see, last column, the values are highest in this column, where only the vermicast has been used. So that has increased the survival rate, and the reason only behind that thing is that the vermicast is very rich in the nutrients. So now moving to our own, the other day we visited there the biochar kiln, and how the biochar is very beneficial to agriculture and what is its role as a carrier. Before going into those details, you must learn here that what are the attributes of biochar, what is the response, and what are the outcomes. In this slide, I have tried to explain here that it supplies nutrients: potassium, magnesium, calcium, phosphorus, and ammonium. When you enrich its content increases. When you blend it with certain organic materials or fertilizers, then the content of those nutrients will definitely increase. Otherwise, sole application of biochar in the soil will create stress, because it absorbs the native nutrients into it, and it will result in the reduced plant growth. In many of the papers, sole application of biochar is not recommended. Always we have to enrich it with microbes, or with vermi wash, or with vermicompost, compost, farmyard manure, or directly we can also add small amount of 2% nitrogen and 1% phosphorus also. But it is rich in potassium, we need not to add potassium, as discussed earlier. Then enhanced water holding capacity, that is the significant attribute of biochar. Even 70%, 80% higher water holding capacities it has. Then changes soil pH, it has higher electrical conductivity, lot of ions are there. It changes the pH as well as the electrical conductivity of the soil. And then when we add the biochar to the soil, what it does, it has very high surface area. You see, these are the pores. Actually, when we heat the biomass at 450 or 500 degrees Celsius, the entire amount of organic substances that are labile to heat get converted into gaseous form, and that gaseous form is released. That gaseous form will again be burned. Those are syn gases, actually inflammable, they burn and further increase the temperature. Then what is left is the structural remnant of the plant. ये जो आपको दिख रहा है यहाँ पे, maybe in the next slide it is better, ये this one, these are the structural remnants of the plant cells. जो सेल वॉल वहां पे सेल्यूलोस वगैरह कार्बन जो भी है वो रह गया, and that is highly porous substance. That is why the plant microbes interact with the biochar in a more efficient way, because of its high surface area and because of its porous nature. So it seems porous like this, like these are cells. It is because of the structural remnants that are left after we heat the biomass. Then high surface area and porous structure provides better habitats. Low temperature biochar provides ready available carbon as energy source, and release of microbial inhibitors, and then control of potential pathogens. Then we have the outcomes. It results in the enhanced microbial community, abundance, activity, and diversity, facilitates biochemical cycling. That is the outcome. Improves soil structure through microaggregates, improves soil enzymes, contamination degradation. This is if a soil is contaminated by a heavy metal, for example, or by some certain organic substances, then it has very high adsorption capacity. It absorbs on itself those contaminants and poisonous materials. Then it results in the lesser effect of the contamination, and transforms those substances into less toxic substances by reduction and hydrolysis. Then contaminant immobilization, it also results in that contaminant is no longer available to move in the soil. it immobilizes it, and this results in because of the absorption onto the cells and formation of complexes and precipitates, and finally these all results in the promotion of the plant growth. So there is a fundamental theory behind the application of biochar. Then there are reports that trichoderma has been loaded onto the biochar materials. Then it has been tested in certain plants such as tomato and has given some favorable results as reported by Sani 2020. Then this is the plant microbiome interaction. When you add microbes or to the soil, or the microbes that are favorable are already present in the soil, or certain PGPRs, then those release flavonoids, then certain cells or cysts or spores are immobilized on the biochar. Then when conditions are favorable, those immobile cells or cysts or spores will again start multiplying and provide a better habitat for the survival of those microorganisms. So that is the fundamental idea behind the use of biochar. Biochar as carrier materials. Then we have wood biochar, we have compost also, we have zeolite, peat, clay, rice husk, rice bran, rice husk, or those raw organic materials can also be converted into powder and after sterilization used as a carrier. Then kaolin, bentonite clay, mixture of soil and charcoal. Then why biochar? Because of has been reported to increase crop yields. Then blends of biochar with synthetic fertilizers, compost, or any organic material improves the efficiency of biochar. Then biochar has been reported as efficient carrier of fertilizers and biopesticides. Further application of biochar has been reported to induce disease resistance. There are certain papers where it has been reported that it induces some kind of resistance in the plants and unfavorable environment in the soil for the pathogens to multiply. Then all these aspects of the biochar adds many dimensions to biochar research with direct role of agronomy, soil science, plant pathology, and agricultural microbiology. Then application of one thing I forgot here. Then when the cysts or those active vegetative cells reside inside the porous structure of biochar, it prevents the entry of the grazing microorganisms such as amoeba. Actually, the bacteria are eaten by the amoeba or protozoa. The size of the amoeba is bigger than the pore of the biochar that is present there. It is not possible for the bigger protozoal cells to enter into those sites, and that is how it prevents the grazing. इसका ये बहुत बड़ा है. And then the cysts or pores or vegetative cells can stay there. It provides a surface, it provides nutrient, and it provides a micro environment in those micropores to survive. ठीक है ना सर? So that's why we recommend or we promote the use of the biochar as a soil amendment. Then these are the different materials in Kashmir. Also, we can do some work on this because we have good apple area. Then apple prunings every year we get some raw material from this rice husk or rice chaff. Sir, I would like to suggest here that after sterilization of this rice chaff, we can we can powder it. We can also test this efficacy as the carrier for this in powder form. So because it is abundantly available in our campus. Then some of the students also can take up in pathology or microbiology this type of research where we can test different materials and test them against their efficacy and survival rate of the different useful microorganisms, say trichoderma or rhizobium or azotobacter, azospirillum, whatever strains the microbials isolated. We can test them as the carrier materials and see their efficacy, how they can be used. Then rice chaff, then chinar leaf. A lot of 35,000 chinar trees are present in Kashmir. Every year the leaf falls and it gets wasted. Previously people were burning it to use for the use as charcoal, but nowadays people are very rich. They use gas or power heating or hammam or other fuel, not this leaf. This leaf can be also used as raw or after converting into the biochar. In both the situations, both the forms, we can test it for its efficacy in carrying those strains. So this is a little bit data how much apple area we have and how much estimated amount of apple prunings we are available, and those apple prunings also can be converted into charcoal or biochar. Then survival rates on biochar, humic acid and compost. You can go through this. Here in this slide, it has been found that inoculation of PGPR with different carrier materials had a positive effect on the plant growth and yield. And then you can see after 30 days, 60 days and 90 days, the survival rates in different materials. And you see here the biochar also has shown some encouraging results, and it is almost comparable to the peat in the top slide, in the top table. Then the growth of the test plant was also enhanced when we applied those carrier-based materials. And here the biochar also has shown very good result with respect to the nodule number. Now and the test crop was mung bean, है ना. तो biochar का आप देख लेंगे, inoculated biochar here, the nodule dry weight has 5.77, which is comparable to the compost. AA लिखा है दोनों पे, आप देख रहे हैं? Okay. Then here also some carriers as written in the bottom. Survival of Azotobacter, Crococomon, different carriers 1, 2, 3, and those carriers are calcium carbonate, charcoal, compost, clay. Then in the second, some change in the proportion has been made and clay has been added in the carrier second. Then here also the addition of charcoal has given some encouraging results with respect to the survival of these bacteria. And then biochar as azoospermum. Here also in this slide, the studies on the biochar carriers produced from the acacia and coconut shell for azoospermum lipoferum inoculants have revealed their superiority over lignite. In this case also, there are a good number of papers which you can, if somebody is interested in this case, he can go through and see how charcoal, biochar, or these kinds of material are comparable with the other lignite, peat, or even you can test it against the liquid formulations also. But again, when we add some organic matter to the soil, that is beneficial, whether it be raw or after converting into char, that is beneficial rather than simply adding some liquid. Okay, na? So that was all about it. If somebody has some suggestions, some clarifications, can ask, and then we will conclude it. आप सोच रहे होंगे लास्ट दिन में इसने परेशान कर दिया यहाँ पे, हैं? You might be thinking.